It's a special honor and pleasure to be here, for two reasons and more, then as a true pioneer of the off-the-shelf cell therapy, it's one of the major topic of what I'm going to talk today. So be here means something special to me, myself. And also the type of cell I'm working with, you heard the term NKT, and I will tell you, a few other more, we are also working with, belongs to this unconventional T cells. And we do have another pioneer working on that, Dr. Mitch Kronberg sitting over here right in this room, and many other labs at this institution, the La Jolla area, are working, on the type of cells and this area and the cell-based therapy. So it's a true pleasure over here. Today, I'm going to talk about what my disclosure and then cancer immunotherapy, right? So cancer is obviously a very significant, health challenge, for U.S., for the world, and cancer immunotherapy becoming the fifth pillar of standard cancer care, especially starting in the last 15 years. And among those, there's two, outstanding breakthrough, and one of that is cell therapy, cell-based therapy, right? And especially CAR-T, chimeric antigen receptor, engineered T-cell therapy. So far, there are 7 FDA-approved CAR-T cell products. It's revolutionized the treatment, of a couple of blood cancers, and there are more, in the pipeline. You check the clinical trial, .gov, there's over 2,000 clinical trials testing various kind of CAR-T, therapies nowadays. And here's a positive side, but here is the daunting side, right? So far, there's not successful yet. There are promise, but not very clear, a kind of a successful example. There's nothing approved yet, for treating solid tumor. And all the approved ones, including the 7 I mentioned to you, it's all autologous, therapy, which means patient being treated one by one, and as shown over here, that you need to draw blood, you need to engineer the cells, and the entire, effort will only serve that single patient, right? And for the reason, for immunology, we talk about this mismatch, of the MHC, and so that you cannot just freely transfer a T cell from one person to another without causing, problem, okay? And all of those associated with this high cost, right? So the current, this is a lower number, actually, 300, okay? So if you look at all the price, for the 7 FDA-approved cell product, it's ranging from 300 to 500, okay? So it's very, pricey, cost, and it's labor intensive, it's time consuming to make. It may not work for everybody because the manufacturing may fail, or, for some cases. For all of those, you are thinking about, based on the promise and also for the challenge, is that something we can do to make the next generation of cell therapy, right? So we will hope that can be happening. And we chose to, look for new type of T cells. Maybe there's something beyond the conventional T cells we can look at. And we want those cells to be potent. We also want the cell to have this off-the-shelf potential so that we can move away from this autologous therapy limitation, right? So my lab at UCLA here is exactly one major direction we're focusing on. We are trying to develop off-the-shelf, immune cell therapy by harnessing a special, group of T cells. And what are those T cells? So as mentioned, that all the current CAR-T cell therapy based on this conventional, T cells, right? When we talk about the killer T cells, when we talk about the helper T cells, those are what we know, mentioned T cell most of the time. And they are predominant population in blood. They comprise over 90%, of the T cells over there. They recognize protein antigen, right? They recognize protein antigen, breaking down into pieces, and then load onto this presenting molecule called the MHC molecule. And that's exactly where the mismatch can come from. That is why every single person have a unique set of MHC molecule. T cells from that person can only match into those MHC. You just randomly give to another recipient that what may cause problem. That is the root of the problem. However, beside those, there's another group, quite large group actually. So by cell type wide, but number wide is small. Okay, so all of those comprise less than 10%, in the blood. And you look into that, the RT cells recognize antigen beside the protein antigen. And it turned out our body, our cells, all the possible, pathogen, invading pathogen antigen, our immune system should monitor. It's more than protein, right? So, for instance, this NKT cells I'm going to focus on talking about today. The name, people call it invariant natural killer T cells, or NKT cells, iNKT, NKT cells. They recognize something not protein. They recognize lipid antigen, right? Lipid is also a pretty, big compartment, compose our body and the cellular compartment. And those lipid antigen can load onto, molecule similar structure, like MHC. They are called CD1D molecule in this case, okay? And here is a, catch. CD1D molecule, unlike the classical MHC molecule, it's non-polymorphic. So it's one single CD1D molecule in me, in you, everybody in this room and across the world. It's the same one. Okay, so what does that mean? That means like the type of T cell, NKT cells, if we can make those cells as a therapeutic cell carrier, it's safe to transfer between people because there's no mismatch. We do have the same CD1D. Okay, keep that in mind. And beyond that, this unconventional T cell, especially NKT cell, they are multifunctional. So they are born as a memory effector type. They are ready to fight already, okay, if you can make them. And they carry on their surface this unique T cell receptor, can recognize something, it's CD1D matched, some tumor are already CD1D positive, okay, so that they can tackle it already. If you give a CAR, you automatically have like two, weapons already. And remember there's a name NK, in their name, for reason, because they also express tons of those NK cell, fighting, weapons on the surface. So like altogether, see, they are equipped with multiple, weapons and can attack a tumor with multiple, mechanism. And that's quite important because that the immune evasion, right, is something like really a kind of a tumor play, trick to get away from the immune attack. If you can tackle the tumor at multiple front, and then you can greatly reduce the possibility for the tumor to get away, okay? So, and also, this CD1D molecule, turned out to be not only, can be used to recognize tumor, okay? It can also be used to recognize the myeloid compartment in the tumor microenvironment. And those, it's important too because tumor microenvironment, oftentimes provide this immunosuppressive, signal to block the immune attack, and make the tumor cell, to get away from the attack. And for those, CD1D turned out to be a biomarker. And I'll show you later on, like what cell in that environment express this and can be a target of the NKT cell, and which collectively can work together, to suppress tumor growth. And those unconventional T cell also different from the conventional T cell in a way. They are really like to be tissue residential. Okay, so conventional T cells, their major job, they survey the body, they really circulate, kind of, that's why you probably see a lot of them like in the circulation. But it can be different, like for a tissue, okay? And unconventional T cells, including NKT cells and others, they take residence in the tissue. They already very good at go to tissue, infiltration tissue, and stay over there until their job done. Okay, and again, that may be important, especially for the solid tumor and the other situations. Okay, and because they are safe to transfer across people, that gives them the off-the-shelf, possibility. Okay, so talking about all of those good things, right? So here is the daunting part. Like, they are low number, right? We mentioned that low number, but for NKT cell, that number is particularly low. Like in human blood, we are talking about around 0.1%, of those cells. Yes, you can try to grow them out, from the donor, PBMC, and grow into a cell product. It's challenging, right? And it involves the long culture, and you may risk of exhaustion during the expansion, and the purity is always, a challenge too, because you expand them from a small number, from those many, many more number of the conventional T cell. Any contamination of those conventional T cell in the final product may risk, causing, the problem like GVHD later on, and you want the purity to be really high, and that's technically challenging. Okay, so how do we deal with that? My lab in the past over a decade, it's through 3 generations of Ph.D. students, like really devoted their time and do the hard work. I see some of the undergraduate students, joined in this study before sitting in this room now, kind of work here at La Jolla region. And our idea is, why not we think a little bit differently? We harness the power of stem cell, right? So, and the ultimate stem cell give rise to all the blood cell, including T cell, called hematopoietic stem cell, right? Another name is the blood stem cell. How about we engineer those stem cells, and then we program those cells to become NKT cells, right? And we program it in a way to be quite effective, and we will program in a way to be very pure, like the only committed path is to NKT. And then we can achieve number, we can achieve purity, right? And because the stem cell, especially hematopoietic stem cell, really have good ability to accept gene engineering. We can think about the gene engineering we give to it to make the most powerful and suitable, CAR-NKT cells for the use. And I'll show you like what we have done and what the outcome, look like. So here is the technology platform now. We chose to use cord blood as a resource of the hematopoietic stem cell because cord blood is good commercially available. There's a stable, available supply chain existing, to obtain, those cells. And there's always baby to be born in the world. So the cord blood, a kind of a resource is a, quite a reliable resource. One single cord blood donor, gave a range of number, of those hematopoietic stem cells, use a marker identified as CD34, okay? And averagely, it's around get 5 million, of those cells to start with. Is that enough, for a therapeutic dose? Absolutely not, right? We need to think about a massive expansion, from that source to the outcome NKT cells. So we have devised this culture. It's ex vivo. It's HSPC-derived NKT cell culture. It's a streamlined 6-week culture. Okay, it's feed-free and serum-free during this major differentiation, step, and we incorporate the gene engineering step at the very beginning. Okay, this will also help us for practical wide because we are dealing with a very small number of cells to start with. We chose to use one single lentiviral vector, deliver the essential, programming gene to be there. Turned out to be the T cell receptor gene. The iNKT T cell receptor gene is the key gene. If we give it to the hematopoietic stem cell early enough, and then we put the cell into this culture, driving them down the path of T cell differentiation. The transgenic iNKT T cell receptor will give the instruction to make sure that the cell become an NKT cell at the end. Okay, and because the gene delivery capacity of lenti-vector, we can co-deliver other cargo genes. And of course, one favorite cargo will be CAR, right? So like we put chimeric antigen receptor gene over there, then we can direct the cell product to target, whatever the disease, indication, in this case, the cancer, type. And we still have space over there to incorporate additional, genes. And why not think about give something immune regulatory enhancement gene really can help the NKT cell function to fight tumor. And that's a group of candidates you can consider. And one of the ones like we, elected to move first forward called IL-15. IL-15 is a cytokine NKT cell really like. I guess NK cell also really like IL-15, right? But giving it to them, and then the cell will carry their own enhancement genes and really help the product to perform. Okay, and the question is that do we need to design this culture process and engineering differently for every single, disease target we change? We want to target this tumor, we change CARs. Do we need to redesign this whole thing every single time? Fortunately, the answer is no. Okay, once we set up this whole culture process, we only need to change the lenti-vector. And the culture is the same. We tried a bunch of, the CARs we can cover many, blood cancer target as well as the known target for the solid tumors, and we can successfully make all of those CAR-NKT product. Okay. And here shows that, stepwise development. We showed like if we put NKT, transgene delivery into the system, we can grow out very pure NKT cell over the 6 weeks. If we co-deliver a CAR, we can grow NKT cell now co-express CAR. Okay. And if we give IL-15, in addition to that, we can grow CAR-NKT cell co-express IL-15. Okay. And the product is very pure, as you can see over here. All the cell outcome, there's no need to incorporate any purification step either at the end or in the intermediate step. It's a one stream of the culture. Okay, we isolate the cord blood CD34 HSC, do that one time of the gene engineering using lenti-vector, and we dump into this culture. 6 weeks later, collect the entire product. And the product can be cryopreserved, and they survive cryopreservation and recovery pretty well. So like we just collect everything, and you can freeze down, and then that's the cell product. So all the characterization I'm going to show you behind this is based on the cryo-recovered product. Okay, very well. If you look deeper into the, genomic level and molecular level about this whole differentiation process, here is if you're tracking timeline, for the single cell RNA-seq analysis, you can see, the cell, CD34 positive HSC has many potential at the beginning to become all kind of blood cell. But along the culture condition define them to go down the T cell path, and they become this very pure population of the T cells at the end. And the product-wide, I told you it's pure, but is that really like the most, important thing is that we do not want any of the randomly recombined conventional T cell receptor to be there, because that gives you the risk of GVHD. And here is for the single cell level of the TCR, sequencing at the genomic level, and we can show that the product indeed really pure. We didn't detect the randomly recombined TCR. The only TCR to be there is the TCR we give to them at the beginning. It's encoding this NKT T cell receptor. Okay, and then here is the whole genome, the deep RNA-seq analysis to study the nature, what exactly whole, like 22,000 gene, analysis, like how they look like. And the positioning over here, PCA plot, show them far away from NK cells. So they are indeed, at the T cell side. Okay, and along the T cell side, if you look at which one they most closely resemble, NKT cells. Okay, and then the phenotype of that, if you look at that, they are pretty typical, NKT cells. Okay, they express TCR, they express, the co-receptor, and then they express this very, potent effector function phenotype, which means they secrete a lot of effect, cytokines. They make a lot of cytotoxic molecules. So they are powerful killer and the effector cells. Okay, mechanism of action. I mentioned at the beginning that the one thing we like those cell carrier because they themselves, remember, they have this T cell receptor, can recognize the CD1D positive target already. And that's a group of myeloid lineage and the B cell lineage of the blood cancer. They do express CD1D. That's a few of those solid tumor, express CD1D. Okay, and then plus the CAR, that's already two weapon, to use, right? And those cells express tons of those NK, receptor can recognize the NK ligands, which abundantly expressed on almost all tumor cells, especially on the solid tumor cells. So that can be a third, group of weapon. So that's three mechanisms over there. And some tumor, even though they are not CD1D positive, that they still have CAR, they still have those NK, ligand to do the job. So here that take one example as multiple myeloma, cancer patient that we collect the primary patient sample, and then we analyze the surface markers. Indeed, in this particular cancer type, we find all three, mechanism will work, right? Because the tumor express the CAR-NKT, in this case BCMA, and they express CD1D, they express, a lot of those NK, receptor ligand. Okay, and then here shows that in the combination of that, make them actually have certain advantage compared to pure CAR-T cells because the additional weapon and this really ready to go effect memory and powerful killing function they already have. Those are in vitro killing, right? So whether those cells can perform in vivo and do the job right. And here is that the model we use for modeling the human multiple, myeloma, NSG mice, human multiple myeloma tumor cell engrafted. And here, we label the tumor cells with luciferase, so that you can visualize it using the live animal image, right? So whatever you can see, light up over here, those are the tumor. And this pretty faithfully model the nature of multiple, myeloma, because you can see all tumor go to the bone. They will live in the bone marrow over there. Okay, and here is that by monitoring that, we can, measure the efficacy of the tumor killing. And I would just point out to the conclusion over here. So we side by side compared the conventional, BCMA targeting CAR-T cells. It's over here, the green, kind of a color and the line. And then we have the two type of the product from the CAR-NKT, being compared over here. One is without L15 enhancement in blue, and the one with L15 enhancement in red. And the one with the enhancement perform the best. Okay, and in that case, it's really a kind of a clear tumor, and leave the mice, live long enough. And CAR-T cells, in this case, didn't clear everything, so, and the mice kind of die in a mixture of both, residual tumor growth as well as GVHD. Remember the GVHD risk we talk about, that's prevent the conventional CAR-T to be off the shelf, and here is clear demonstration of that risk is indeed true over there. Okay, how about the therapeutical cells, right? So where are they? Can they really as mentioned, they have this tissue infiltration, residential, they can find a tumor and do their work, right? Is that indeed the case, right? How do we see that? And we adapt the same, in vivo model, but we use a kind of a mirror imaging approach, okay? Remember that what I just showed you, I mentioned that we label the tumor cell, whatever light up a tumor cell, right? So in this particular experiment, everything is still over there. Okay, tumor cell, therapeutic cell is there, exactly the same dose, same injection route, and the same timeline, okay? The difference is now we label the therapeutic cells. We label the CAR-NKT and the control CAR-T, cells over there. So whatever you see light up over here, those are the therapeutic cells. So what do we see? Okay, this allows us to tracking persistence. This allows us to tracking the homing, right? This also allows us to tracking whether we see a clonal expansion of those therapeutic cells after they see, tumor over there. It's very, very important why I emphasize this. If there's one single biological correlation to predict whether a patient receive CAR T-cell therapy will be successful at the end, really benefit from that therapy, is clonal expansion. If you see the CAR T cells in that patient persistent clonal expand, you see a beneficial outcome. You don't see that, you don't see, clear, there's no clear, prediction at the end. Do we see the clonal expansion? Okay, so I want you to focus on this red line over here. Remember, this red color indicates the L15 enhanced CAR-NKT, which is the preferred, a kind of a design of the CAR-NKT, product. You see a very beautiful clonal expansion. That associated with, remember, going back to the efficacy, that shows the very clear clearance of the tumor, right? Okay, and also important, after the tumor cleared, if the cell non-stoppingly just proliferation and the hang around, that's a problem, right? You worry about the safety. They do calm down after the tumor clearance, but they're not totally gone, okay? So there's some hang around, over there. So that is the stage over here. You can see the time course. Even at the very end, you see some residual cells still hang around really long, Those are kind of long-term memory, they preserve over there. And for some of these mice, we tried the secondary tumor challenge. We give the tumor cells again. The remaining cell respond again. You see a second, wave of the clonal expansion. So just to show you the design, allow them to persist, clonal expand, form a memory, and have this long-term, ability. And where are they, right? Here is an enlarged, version to show you where they are. In the bone marrow. That is where the multiple myeloma tumor cells go, right? So they do know where the tumor are. They do know, go there. And they do a clean job over there. Not like the CAR T-cell, that's what CAR T-cell do. CAR T-cell go to the bone marrow, do the job, and then quickly, they mount this GVHD, start to attacking everywhere. That's why you see this entire mice is light up, over there, and by the time they almost, a kind of have a death sentence, to go down the way. Seems the cell, they show good efficacy. The kinetic study shows that they indeed, home, persist, clonal expand, and do the job, as expected. And there's a lot of more, further detailed gene profiling we performed to fully understand at the genomic level, molecular level, like what's going on, why we see the phenotype we see, right? So like it's, we collect the samples from the experimental mice, sequential wide along the timeline, and then we fractionate the cells, and then we perform the single cell RNA-seq. So like we're able to tracking, what happened to the therapeutic cell and what happened to the tumor cell along the way. And here is just a combined analysis to show you like we can grossly, for instance, collect the therapeutic cells, split them into the different stage, right? There's a proliferation stage, resting stage, there's a effect memory stage, that's more exhausting, like not so functional, stage. And those can be all analyzed and compared across, the different type of the cell product. In particular, we want to compare what happened to the CAR-NKT product versus the CAR-T product, and across the different, organ and the region. So those in tumor site, okay, data shows over here. I probably point your attention to this last, a kind of a pie structure over here. And here is a CAR-NKT, here is a CAR-T. And you can see a quite enlarged subpopulation number, I believe number 2, right? And what are number 2? You read it over there. Those are the effect and the memory population. So those is probably the reason, give them this quite, powerful efficacy readout. Okay. And more analysis, the dynamic, analysis show you that indeed, if you look at the transition between the different stages, and the CAR-NKT cell have a ability to hang around at this cluster number 2. Again, it's effect memory, cluster longer. Okay, they can stay there longer, and the CAR T cell have a tendency to slip quickly, into that more exhaustion, a kind of a stage. Okay, and the pathway analysis shows that it's CAR-NKT cells enriched for the proliferation, effector function, all of those good things correlated with their performance, and indeed IL-15 is really helping them, and the IL-15 pathway is kind of enriched, of course, because of that. I mentioned to you at the beginning that the multiple targeting, capacity of the CAR-NKT platform is what we really, like. Because it can have the potential, to counteract with the tumor evasion. Is that indeed the case? Okay, in this multiple myeloma, model, we collect the tumor cells at the very end, okay, after the long fighting between the tumor cell and the therapeutic cells. And we analyze this CAR antigen, right? CAR antigen here is BCMA. So look what happened. So for the blue one over here, that's conventional CAR T, right? You see even in preclinical animal model in 1 month, you see this downregulation of the BCMA in that tumor cell already. And it's no surprise because this is what people see in clinical, in cancer patient, multiple myeloma patient. Okay, and if you look at the CAR-NKT, treated tumor cells, you don't see that. Okay, and we can, imagine that maybe because that tumor now and the CAR-mediated attack and TCR, CD1D mediated attack, as well as this around, a dozen, of the NK, receptor, right? NK ligand, mediated attack, and maybe that's why the tumor have no way to get around. Okay, so the tumor microenvironment, again, we mentioned at the beginning, says that the unconventional cell, like NKT cell, that they have a way to modulate and reshape the tumor microenvironment. Why? Through whatever they can recognize, right? And what NKT cell recognize? They recognize CD1D molecule. So if we fractionate the tumor microenvironment and see what cell express CD1D, then we may identify the target, of the NKT cells, and we'll see whether those cells even matter or not, to an anti-tumor immune response, right? In this case, we take the, again, primary multiple myeloma, patient sample. We use flow cytometry, fractionate the cell, and understand what's, other cell composition. And you can find, many of those. You will find the conventional T cells for it. You will find the NK cells. You'll find the B cells. You'll find the, a lot of them myeloid cells. You'll find the granulocytes. Okay, so if you look at CD1D expression, there are two populations really standing out, express a high level of CD1D. What are those? One is tumor-associated macrophages. Another is myeloid-derived suppressive cells. Those cells are the, culprit, like in the tumor microenvironment, they are the one really kind of provide this major immunosuppressive function, right? So there's many, cancer immunotherapy strategy trying to tackle that part. And guess what? The CAR-NKT cells through their T cell receptor, those are their natural target. Okay, so do they really can do anything, even after they see that? So if we mix the primary samples with CAR-NKT cells, overnight, it can happen as quick as 12 hours, actually. Okay, but if you analyze at 24 hours, this is what you see. CAR-NKT cells very effectively deplete TAM, deplete MDSC, but leave the other things untouched. Right? So you can preserve the other anti-tumor immunity, but you make this, major immunosuppressive gone. So this looks promising, but again, it happened in a dish. It's that indeed in a, in vivo, in a tissue, right? The NKT cell can still, kind of navigating around, find the TAM, find the MDSC, and do that job, okay? So we look into the in vivo model, the same NSG model. So we give tumor, give therapeutical cells, at the end collect the whole, bone marrow compartment cell back, and then we use single-cell RNA-seq to fractionate and read out like what happened over there. I would like to point your attention to those clusters, number 4, 5, and 6. Okay, and 4 is M2-like macrophage, so they resemble the 10. Okay, and 5 is interesting. It's a CRS related macrophages. Those are the ones gave you the side effect, like a CAR-T cell. In therapy, gave cytokine, release syndrome. Okay, and then the 6 is MDSC. So blank NSG mice, this is a basal level of, they have a little bit of something. It's not really tumor associated, but it's kind of the residential, macrophage over there. After tumor challenge, without any therapeutic treatment, you see a huge enrichment of the myeloid population. Okay, that's 4, 5, 6 population. Tumor induced all of those, we all know that, and that help tumor to grow. So they are kind of, work together over there. Look at what happened to CAR-T. CAR-T cells have all the ability to kill tumor, but they don't do too much, to this microenvironment at all. This entire, myeloid population preserved over there, non-touched. Okay, look at what happened to the CAR-NKT group. It's basically gone almost. Okay, and the fractionate and look at its quantification, and we validated using flow cytometry, and it's true. You see that after the CAR-T cell treatment, the TAM is gone, suppressive, MDSC is gone, and the CRS-related macrophage is gone. Okay, so what does that mean? Does that mean that maybe this can be a beneficial thing to control the side effect, to reduce CRS, because that's a major, side effect of the CAR-mediated cell therapy? Okay, so is that indeed the case? So in the model, so we measured the CRS biomarker, right? Okay, so it's CRS mediated by CAR-T cells stimulate myeloid. Myeloid make all those inflammatory cytokines. There's a group of them, and the most significant one, well characterized, called IL-6, right? So like we do see that compared to the blue bar over here, those are CAR-T induced tons of the IL-6, and the CAR-NKT actually much reduced, right? That IL-6 level, another called SAA3. That's another biomarker for that. Can we read out a really phenotype-wide, functional wide, read out some consequence if we reduce that CRS, reading? Is that indeed something can be done, to a living animal in this case, and maybe, a kind of a patient in the future? So we use that need a little bit more than just NSG, model, because you do need a functional crosstalk between the therapeutical cell and the myeloid cell, which means you need to give both compartment as a human, right? So like we need to reconstitute the human myeloid cells in the mice, in order to read that phenotype out. And we did, okay? So by adopting a advanced humanized mouse model called NSG SGM3 mice, okay? So those have the human cytokine supporting the maintenance of the human myeloid cells. And we take that mice, we gave the human CD34, HSPC at the beginning in the mice to reconstitute the mice. Now we have a humanized mice, have this full compartment of the human myeloid cells. Okay, now we can read. Now if we give a tumor and then we give our therapeutical cell, we will be able to read out the real, meaningful interaction and the consequence of that myeloid compartment modulation. And here is a very striking results. in this case, this one we use CD19 CAR-T, compared with a CAR-NKT. Okay, look at the survival curve over here. The CAR-T cells kill tumor. In this case, it's heavy tumor load. They also induce very, very strong, CRS side effect in 3 days. Those are hours, by the way. Okay. In 3 days, it kill the experimental mice. Yes, tumor are gone, but the animal, also died because of the very severe, side effect. But none of the CAR-NKT treated mice died. Not only they don't die, if we try to score it, so the CRS also reflect as, fever, reflect with body loss, dehydration, and all of those, symptoms, if we can score them, it's very clear. Like those mice, they look normal, they move around, they eat normally, drink normally, they don't drop body weight. They seems to be totally fine in this case. So we're happy to see that. And if we collect back the bone marrow cell at the end and look, okay, and this system, by the way, besides reconstitute a human myeloid cell, also reconstitute human B cells, right? So if CD19 here, human B cell is a target, you can see CAR-T cells, no problem, deplete all the T cells, B cells. But look at the myeloid cells, perfectly stay over there. Okay, then that's the resource, of the CRS and what makes those cells die at the end. And look at what happened to the CAR-NKT treatment. B cells are gone, myeloid cells are gone. So there's no myeloid cell making IL-6, making those inflammatory cytokines. The CRS symptom, the side effect greatly reduced. Okay, so there's another side again. Even though I promise you there's no GVHD risk, don't worry, it's the same CD1 molecule. We still need to really read out to see indeed the case. Okay, are those cells somehow recognize, the tissue, the mismatched tissue in a different way. Okay, and that's two classical, assays we performed. One is this in vitro, mixed lymphocyte reaction assay. So we just mix the allogenic off-the-shelf cell product with donor mismatched random donor PBMC and culture them. Okay, so if the cell will recognize the mismatched donor cells and will respond, and then interferon gamma will be released and it can be measured. So here shows that CAR-T, of course, only the matched pair won't really make interferon gamma, and the mismatched one show different level of the miscompatibility, and then you can see all those interferon gamma production. And for the many, we showed 5 over here, but in reality we did over 20, whatever the collection we have for the PBMC, collection in our lab, and it's very clear that those cells don't really mount a mismatched, a kind of GVH response over there. And in the animal model, NSG mice, as mentioned, the mismatch and the recognition cross species, between CAR-T and NKT and NSG mice, that we can see that the pretty much, get screwed up, and all of those tissue infiltration and destruction. Okay, and then the NKT cells, it's quite safe. The mice stay alive, we keep the longest one as over a year, until our animal facility told us we have to kill those mice, but they are quite healthy, kind of until then. And the HVG, risk, right, off the shelf, that's another thing I want to worry about. Can they be rejected by the host immune system? That's not a safety concern, but it's an efficacy concern, right? If they are rejected immediately, they cannot really do their job, kind of good enough. And here is that the various, assays that we performed over here, and this can be T-cell mediated rejection, and can be mismatched NK, mediated rejection. And here is that the assay, mixed lymphocyte reaction assay, to assay the T-cell rejection and NK rejection. And I simply want to point out that compared to the conventional CAR-T, that CAR-NKT cells should reduce, a kind of the HVG, risk, okay? And that correlated with what T cell recognize and reject is through the mismatched MHC molecule, right? So turned out those CAR-NKT cell express reduced surface level of the MHC molecule, okay? They reduced for the HLA-1, molecule, and almost like down-regulate the HLA-2, molecule to the minimal, undetectable level. And that sustained not only after we grow the cell as a cell product, it's also sustained after, this whole tumor fighting in vivo. In a month, they still remain that, low immunogenicity. And why, right? So like, is that a protein level? Is that a gene level? We look at the messenger RNA, and indeed, we find it's low, expression of messenger RNA encoding MHC molecules. And then for NK, they recognize the NK ligand, right? So it's all pretty low, on the NKT cell. And we further, look into it, and shows that come from a combination effect of epigenomic modification of the cell product that they already like hypermethylate, many of the region encoding those mismatched reading, molecules, including the HLA, including the NK, a kind of a receptor ligand, okay? And moreover, okay, so this signaling pathway also control the upregulation of those molecules during inflammation, especially after interferon gamma induction. Okay, so we look at the interferon gamma signaling pathway. We found that the CAR-NKT cell produced by our method seems to have a desensitized interferon gamma signaling network. And it's go down to, the phosphorylation of STAT1, which is the core, of activating entire interferon gamma, network. And that one, is really compromised, the phosphorylation, okay? So it seems that's the reason why it keeps them stable, even during a fight, during an inflammatory environment, after a long interaction with tumor cell, they are able to maintain the low expression of those. Okay, so is that just for one product? I mentioned to you, we are able to incorporate many, different CARs onto this platform, and we tried the different designs, tried the secondary generation, third generation. We tried the different signaling domain, CD28 domain, 4-1BB domain, and it seems that it's all tolerable. So like it's quite flexible and versatile, platform. And by to prove that we incorporate many, you probably, some of you are working with or familiar with some of those markers, like directing to target some certain blood cancer or solid tumors, and we are able to successfully make the cell product with this high, efficiency. And I would like to mention about the yield. So from one single cord blood donor, remember we mentioned it starts with this very small number, like 5 million, HSC. We can expand the cells for 1 million fold, at the end. So which gave us a trillion, like 10 to the 12 level, of what does that mean? For the CAR-T, therapy or cell therapy nowadays, like 10 to the 8th to 10 to the 9th is usually the range, for the dose. We can make 1,000 to 10,000 dose from one single cord blood donor. So that make it practical to enter clinical and maybe commercialized in the future, right? Okay, so we didn't stop over there. That's HSC. Can we think about to produce an unlimited supply from an unlimited resource in the future? There is possibility, right? The iPSC, which like Dr. Don Kaufman, has been really a kind of a pioneering for differentiation, iPSC, down to the mature and functional immune cells. And we tried that, okay? So we can start with iPSC, and we can differentiate iPSC to HSC, and then down the stream to the CAR-NKT cells. And we are able to do that. And we're packing up the results for publication, hopefully, this year, you will see those results. And by doing that, we can open up, to the supply end, right? And is that NKT cells are the only cell, can be think about and utilized, for develop this off-the-shelf immune cell therapy? No, there are more, right? So like Dr. Mitch Kronberg sitting over there, and it's really expert pioneer study made cells, mucosal-associated invariant T cells. And those are very powerful cells and interesting, and they repopulate the mucosal system, can be think about, to tackle, diseases in the GI tract, mucosal system, called cancers, autoimmune disease. So those are of interest, right? And there's another group called gamma delta T cell. Those are also unconventional T cell, also do not have GVHD risk. They also recognize non-protein antigen, which may be of interest for tackle a series of the, disease, including tumor, especially solid tumor. So all of those, like we are, kind of expanding our research to work with. And it's cancer the only disease so we can target? Of course not, right? Then by targeting, B cell, autoreactive B cell, antibody, all those, we can tackle the autoimmune disease, which the autologous CAR-T therapy, have been pioneering right now, right? Okay, and also that's infectious disease. If we find the proper antigen, define the, CAR, define, we can, tackle those diseases too. So it's a decades journey. Take us long, that it's 10 years, again, the 3 generations of the graduate student and many undergraduate students, and we are here. And we think we are at the stage already, to really to move into clinical and really test the real potential, of this platform. And we are having the IND, enabling study going on, and the 2 lead product are moving on right now. For the blood cancer, we're targeting multiple myeloma, as I showed you the majority of the data for targeting BCMA that can also extend to treat the autoimmune disease once the product is made, because that is off the shelf. It will be able, it's off the shelf, it's allogenic, it can be treating, any fitting patient. And for the solid tumor, like we have a couple of candidates, lining up, but the methicillin targeting one is the one like probably move the earliest. Okay, I'll stop over here thanking my lab, many, generations of the trainees, and the current lab members, for making this possible, and my collaborator, my funding, support. And I'll stop over here, and I also thank you, for your attention, bearing with me in this early morning, seminar, and I'll be happy, to answer any questions you may have. Thank you.
A lot of interesting things, a lot of ideas. You find your cells kill off the myeloid cells, the MDSCs and such,
yes.
Right? But so this is sort of like the NK cells, right? So NK cells don't go around killing your own normal cells.
Right.
So, and presumably normal NKT cells aren't going around killing all these myeloid cells, which have high expression of the CD1.
Yep.
So is it because you're adding the IL-15 or something that these are more active that they're killing those myeloid cells? Or, why does that happen, right, as opposed to what's
then that's a very, very good question. You would worry if you give, a patient the cells and they will wipe out of their myeloid cell. That's not good, right? Because we do need a macrophage, do their normal job sometimes. Okay, so I can give you data from this particular model, like we are studying. So we see the depletion of the myeloid cell in tumor microenvironment. We don't see the depletion of the myeloid cells in the non-tumor tissues, including the peritoneal cavity, and including spleen, including lung. We don't see that. Okay, I can think about 2 reasons why this happened. One is that the CD1D expression level. Okay, see, CD1D confined to express normally in myeloid, compartment, but it's only significantly upregulated when the myeloid cell fully differentiate and enter that inflammatory or activation stage. So if you compare to the CD1D level, that quiescent and the tissue residential one, they express low level. Okay, only like this tumor interacting one, that one express sky high. So that can make a cutoff maybe, so kill or not kill, right? And another very important reason, location, right? So like the therapeutic cell, by giving a CAR, they know where they go. In this case, they go to the, multiple myeloma residential, bone marrow area. Okay, we did the ovarian cancer that confined in the peritoneal cavity. Then they go to peritoneal cavity. So I guess it's a combination of the detection, sensitivity to distinguish the fully, a kind of differentiated in action, myeloid cell versus the quiescent staying, tissue residential and maintenance myeloid cell. Also the location, of the therapeutic cell will make a difference.
Beautiful work. I'm curious about, you may have touched on it in the beginning, but is IL-15, do you think that's going to be the most potent, like engineering moiety, or would there be other cytokines or transcription factors that could also have maybe different or more selective effects?
A very good question. And the answer is, we certainly love IL-15, and for many good reasons, and also IL-15 engineered into therapeutic cell, in particular NKT platform, has been tested to be safe in clinical. So, it's powerful, useful, and safe. Then that's probably the reason we picked the first to go. Is that the only thing you can give to NKT cells? Of course not. There's a long list you can think about can help NKT cells. Many are overlapping with NK cells, So but the regular toolbox, everybody are considering, and it's currently in various stages of the preclinical or clinical development you can think about, right? IL-21, IL-18, IL-12, and more, so that's certainly more you can think about.
One thing I'm wondering about is those DCs have very high levels of CD1D also, generally, at least when other people have measured that. So do you see an effect on CDC1 or other dendritic cell types, which might have a counter effect, although it's good that you eliminate the myeloid suppressors?
And that's a very, very good question. We actually worried at the beginning also, wow, it will wipe out of the dendritic cell too. It's missing, something for fighting tumor, right, later on. So look at the data over here. That's population number 2 is DC. Okay, so the color is, okay, so the color is 2 over here. One is spelled. Okay, so we have another way to measure, but basically you can see other things are gone. Those populations are not wiped out as much as the myeloid cells. Why? We wonder, right? So, look into literature, and interestingly, it's saying that, DC as the most powerful APC naturally also makes them a target for CD8 T cells. Why they still can do their job, right? So there's some self-defense mechanism for DC, and including the overexpression, very specifically so unique, just in the DC population, gene encoding an enzyme to degrade granzyme B. Yeah, so that's almost like it's you're shooting missiles and there's something, to just eject the missiles. So those gene, that particular gene, if we plot it onto this single cell RNA-seq data, it very specifically land at the DC population, and that's upregulated in DC after the treatment of CAR-NKT. So seems the same defense mechanism keep the dendritic cell being able to hang around, kind of doing the normal job in a normal immune reaction. It also exists when CAR-NKT cell appeared in that environment. They are able to defend themselves at least to certain levels.